we're going to move into the main meat of our conversation here, which are CMC considerations for these next generation molecules. Maybe we'll start with Morris here. Morris, with these innovations around molecule design and components, whether it's linkers or constructs, how do you think this impacts the way developers think about and approach CMC?
Well, first I would say, previously we were talking about continuing innovation. So a lot of times you're building off of what was done before. So companies will bring forward new ADCs, and maybe what is new is the target, or conversely the payload, and then they'll use other parts that have been previously used. If you are developing a new linker or a new linker payload, then you have to take into consideration the timelines of what's the fastest path to the IND. If the linker payload is new or novel, it may take longer to develop the process and scale it up, as opposed to, I'm going to use an auristatin that's already been in the clinic, or I'm going to use exatecan that's already been in the clinic, because there the manufacturing process has been worked out somewhat, and it becomes more plug and play. But if you're incorporating innovation, then everything about the process changes. The manufacturing process, the supporting analytics, all of that changes, and you have to factor that into your timelines. So that's the first thing that comes to mind.
Thank you, Morris. I saw a question coming here from Christina, and I think it's what you're alluding to, where when it comes to payload, what is the next generation of ADCs regarding the drug product configuration? So, Morris, correct me if I'm wrong, it sounds like the smartest approach, or at least to reduce timelines, is to go with a pretty validated payload or drug part of the ADC. Am I understanding you correctly there?
Well, the advantage of that is you reduce clinical risk, because those in front of you have already prosecuted that in the clinic. The disadvantage is if you're using the same linker payload as companies X, Y, and Z, then when you're going into similar patient populations, they might have resistance to that payload. So we do need new innovation, and that is what leads you to selecting a new payload or a new linker payload, to get at some new mechanism of action that overcomes the resistance the patient may have developed to the prior payload class.
Yeah.
And Tyler, if I may add a few things to this, and again, this sort of centers around some of the nuances that with our technology, but I think the paradigm fits for others is, it is like, if I think about the question, particularly drug product configuration, and I'll sort of think through, right, payload selection, DAR, if you have access to various linkers, how do you put all of this together? I mean, it is like in our case, right, we'll start with free payload screening to find a desired effect, whether with our lead payload combo, 1-2 punch on the DNA damage response pathway. But there's other obviously concepts that you could employ. And then, once you're sort of centered around what you want to achieve with the payloads, then you're thinking through your ADC design and construction, and the hydrophobicity of that molecule. Do you need to add a spacer or some other design in the linker to facilitate that? And all of these things have to be tested empirically as well. And then, if you think about, say, cleavage mechanisms, and certain cells may have, a particular flavor of a cathepsin or some other, PSAR, some other enzyme, right, that you may need to evaluate, that then may not recapitulate in vivo, and so you may see a disconnect there. But you needing to understand all of these parts and pieces is really important then to finally align on that ultimate drug product. Or even I skipped over thinking about DAR and how you might want to change that to facilitate the sort of tumor microenvironment that, at least in cell lines, you're not recapitulating, and maybe an organoid model, may be better for that. But it's a long way of saying that I think there's certain things that you can de-risk, right, and that's where you do want to spend some time, but ultimately you've got to go through that process of finding that that final TPP for that drug that you want to unlock, and you're going to be doing a lot of testing still to get there, and perhaps AI and other things can facilitate, but at the end of the day, right, you're going to need to be validating these things in as many assays as you can without sort of the balance of not all of us are a BMS or Pfizer with large budgets to do these things. So when you're a small company, you have to think through those parts.